everyone. Today we are going to talk about techniques in proteomics. So, unlike the genome, which is the complete set of genes within an organism, the composition of the proteome is in a constant state of flux over time and throughout the organism. So, what does it mean? It means that we have a genome and it doesn't changes and it has a complete set of genes within it. So, let's say we have approximately 20,000 genes. So now these 20,000 genes, they can give rise to approximately 1 lakh transcripts. And these 1 lakh transcripts in turn can make more than 1 lakh proteins in our body. So therefore, when scientists refer to the proteome, they are also sometimes referring to the proteome at a given point in time. For example, the expression of the protein in the body is not same always. It is either temporally guided or spatially guided. Now, what does it mean? Temporally guided is at a given point of time, which means such as an embryo versus the mature organism. So the number of proteins and the amount of proteins which you are going to see in an embryo is not going to match with when the embryo will become a mature organism, isn't it? Because while in development, different set of proteins are needed at that time, but when the organism is fully mature now, then another set of proteins are required. So the organism doesn't need the same set of protein which it used to have 10, 15 years ago. So that means it depends at what developmental time we are talking about. Now, if we are talking about time, let me give you another example. Sometimes it also happens that if you take your blood sample in the morning and you do the gel electrophoresis, find out the proteins which are present in your blood, and if you will take the other sample during evening and you will do the gel electrophoresis, figure out the proteins in your blood, and when you will compare them, you will see that even during a day, which is only at the difference of hours, even then you will find the change in the proteome of your body. So, this is how the expression of the protein is always in a constant state of flux. It keeps on changing. Proteome keeps on changing. Now, let's talk about spatially. Spatially means that the proteome of a particular cell type or a tissue within the organism doesn't match with the another tissue. For example, if you will take out the brain of an organism and liver and kidney at the same time, and you will perform the steps to find out the proteins present in the respective organs, and when you will match those proteins, you will see that the number of proteins in brain and the type of proteins in brain, it is not matching with heart, kidney, or liver, right? Why? Because the need of the organ is different. Brain has a different purpose to do, liver has a different purpose to do, kidney has a different purpose to do. So since their purposes are different, different set of proteins are needed to solve those purposes. So protein expression is divided within the organs, which is space, and they are different at the different point of times in a day, which is time, temporal. So, what is proteomics? Proteomics is the study of the proteome investigation, how different proteins interact with each other and the roles they play within the organism. So, this is a very important line. You should understand this. We just don't say that proteomics is the study of the proteome because why are you planning to do that? So we are interested in learning about proteome because we need to know how our body is functioning. And our body functions when all the proteins which are present in my body, they talk to each other. So this is called as interaction. So when the proteins interact with each other, they finally complete a job, complete a function, right? So the purpose of studying the proteomics is just not to know how many proteins do we have or the other species have. It is also to know which proteins interact with each other. So although protein expression can be inferred by studying the expression of the mRNA, which is the middleman between the genes and the proteins, isn't it? So mRNA expression levels do not always correlate well with the protein expression level. It sometimes happen that you may have mRNA A, B, C, D, but when you will try to find out the proteins A, B, C, D, you may only find A and B. You will never find C and D because probably it never got translated, right? And it happens because if the signal doesn't, if the signal doesn't is not coming anymore, then it will not be translated. So the mRNA expression level, it does not always in sync to what proteins we have in our system. So there is always going to be a mismatch between how many mRNAs I have and how many proteins I have. And furthermore, the study of mRNA does not consider protein translational modification. So when we say protein translational modification, it means that we are going to talk about the mutations which have happened in the protein, but you will only see when the mRNA will reach to the protein level, right? You are not going to study that at mRNA level. So that is why proteomics is important, because there are many things which we cannot do at the level of mRNA. So things like protein translational modification, cleavages of the protein, complex formations and localizations. So the many variant of mRNA transcripts that can be produced, and all of which are key to the protein function. So different mRNAs can also subject to making of a single protein. It also happens sometimes. So this is why you may not, do a parallel comparison of mRNA to mRNAs to the protein. So the first experiments that fit the label of proteomic studies, they were performed in 1975 with the development of two-dimensional protein electrophoresis. However, truly high throughput identification of multiple proteins per sample only became possible with the development of MS, which stands for mass spectrometry, and which was developed in year 2000 and so. So so since then the sensitivity and the accuracy of the mass spectrometry have advanced to the point where proteins can be reliably detected down to the attomolar range. So what is an attomolar range? Attomolar means which is not comparable. It is like 10 to the power minus 18 moles per liter. So now with the help of mass spectrometry, you can detect a protein even in a sample which is like 10 to the power minus 18 moles per liter. You can detect a protein, and that too very reliably, even in such a small sample. So, why I'm saying such a small sample, because there will be cases, there will be days, there will be studies wherein the protein sample which we get from the tissue is very, very less. For example, uh, people those who are studying bones and the proteins and the things which are in the in such a historical sample, or for example, if I'm working with a very small organism like C. elegans and Drosophila. So their organs are small, their samples are small, and if I need to know how many proteins they are present in their body, so I need to have the techniques which have the capacity to read such a small sample. So that is why I quoted that. And various other proteomic techniques have been developed and optimized so far. So now, what are the key questions that proteomics can answer? So there are many. One of them is protein identification. So if we will study proteomics, we can identify which proteins are normally expressed in a particular cell type, tissue, organism, or which proteins are differentially expressed anyway. I can do protein quantification, which measures total protein abundance as well as investigating the rate of protein turnover. And we can study protein localization, where a protein is expressed and or accumulates is just as crucial to protein function as the timing of the expression, and as cellular localization controls which molecular interaction partners are targets and available. Next, what we can do using proteomics is post-translational modifications. We can study them. Now, next is functional proteomics. So, this area of proteomics is focused on identifying the biological functions of specific individual proteins. For example, the classes of the proteins or the whole protein interaction network. In structural proteomics, we can have important insights into protein functions. For example, whether the protein is druggable or not, or if you can start the computational drug discovery with that protein or not. In protein protein interaction, we investigate how proteins interact with each other and which proteins interact and when and where they interact. So, these are all the key questions which can be solved using proteomics. Now, let's talk about the proteomics techniques. Proteomics techniques are divided into two segments. One is low throughput method, and the other is high throughput method. Now, what is the meaning of low throughput? So any technique which will say low throughput means it generates less amount of data. And any technique in any science, if it says high throughput, it means it generates large amount of data. So today we are talking about the low throughput methods in proteomics. So the first method we have is antibody based methods, and the technique is called as ELISA, which stands for enzyme linked immunosorbent assay, and Western blotting, both of them. So they rely on the availability of the antibodies targeted towards a specific proteins or epitopes, or to identify proteins and quantify their expression level. So in this technique, what happens is you have you have a bench, a bed wherein you have antibody which is specific to the protein you wish to uh, you know target. And when the protein comes into the vicinity of the antibody which is specific for that substrate, they bind to each other, and then you send the secondary antibody conjugate so that you can see in which wells the interaction has happened, and you have finally found out the protein of your interest. So this is ELISA based method, which is the first one. So it is a low throughput technique. At once you can deal with 100 or so samples. That's all. So next is gel based method. So two-dimensional gel electrophoresis, called as 2DE or 2D page, is the first proteomic technique developed, and it uses an electrical current to separate proteins in a gel based on their charge, which means the first dimension of the separation, and then in the second dimension they are separated on the basis of their mass. And then there is another technique called as DIGE, stands for differential gel electrophoresis, and it is a modified version of 2D page, and it uses different fluorescent dyes to allow the simultaneous comparison of two or three protein samples on the same gel. So in case of 2D page, you run one sample at a time, but in case of dyes, you can run two to three samples together, provided the colors of the samples are different. So you can compare the different samples on the same gel. So these gel-based methods are used to separate proteins and before further analysis, by example by mass spectrometry, as well as for relative expression profiling. So on the left hand side of your screen, you can see the gels, and this is the gel which has come from 2D page. And you can see these all blue dots is actually the blue dye, and this blue dye is conjugated with the protein, and this is how we can know at which places in the gel we have proteins, but we definitely do not know which one of the dot is which protein. That is yet to be done, and that will be done by using mass spectrometry. Now, the third low throughput technique is chromatography based methods. So, chromatography based methods can be used to separate and purify proteins from the complex of biological mixtures such as cell lysate. So, for example, we have ion exchange chromatography, we have size exclusion chromatography, we have affinity chromatography. So, these are different types of chromatographies which one can use to separate the proteins from a mixture. So the ion exchange chromatography, it separates proteins based on charge, and size exclusion separates proteins based on their molecular size. And affinity chromatography, it makes uses of the reversible interaction between the specific affinity ligand and their target proteins. So, for example, you can have affinity chromatography, wherein you will already have ligands. Those will have affinity towards IgM and IgA molecules. So, since you already know that the ligands you have incorporated in your chromatogram has affinity toward IgM and IgA. So, if you are running the serum from that column, so IgM and IgA are going to bind to the column, and rest of the material will elute out. So this is how it can be used to purify and identify the protein of interest, and as well as to prepare protein for further analysis with MS, mass spectrometry. So now let's talk about high throughput methods. So high throughput methods in proteomic techniques are those who generate enormous and large amount of proteomic data. So one of the technique is a protein microarrays. So protein microarrays, like I said, microarray. So it is similar to gene microarray. It's just on my chip here. I'm not going to have the mRNAs or cDNAs bounded to the chip. Here we have applied small amount of protein sample to a chip for the analysis. And this chip is sometimes in the form of a glass slide with the chemically modified surface, of course. And specific antibodies can be immobilized to the chip surface and used to capture target proteins in the complex sample. So you can see here that this is a chip, and this is the place where they'll be captured. So they have the antibodies in place, and now the analytes are coming from the sample which we are going to pour, and this specific antibody is for a specific protein. So eventually that is going to come and bind here, and then we also have a label who will tell us that we have caught the protein of our interest. So this is termed as analytical protein microarray, and these type of microarrays are used to measure the expression levels. So if we will have, if we will have more sample of particular protein, so then we will see the more color which is corresponding to the labels we have put in for that particular protein, and that is how we will come to know that how much protein has been attached, and this is now we will come to know how much of the protein is present in the sample. So this is also termed as analytical protein microarray, as these types of microarrays are used to measure the expression levels and binding affinities of the protein in a sample. So functional protein microarrays are also there, which are used to characterize protein functions such as protein RNA interaction and enzyme substrate turnover. So in a different type of microarray, which is called as reverse phase protein microarray. So proteins, for example, healthy versus diseased tissue, or untreated versus treated cells, are bound to the chip, and the chip is then probed with antibodies against the target protein. So you can see in your picture here, they have already, have bounded the analytes on the chip, and now they are pouring in the antibodies, and the antibodies are also tagged. So this is how we'll come to know whether the sample has come from healthy or it has come from the diseased tissue. So this is called as reverse phase protein microarray, and the other one was the forward phase protein microarray. So now this is the real image of instrument or a technique called as mass spectrometry. So this is a mass spectrometer, and it is used for for the identification of the protein, individual proteins in our sample. So there are, so the techniques which we discussed just now, they relied on the gel, if you saw that. So there are also techniques which do not rely on the gel method. So there are several gel-free methods for separating proteins, and one of them is ICAT, stands for isotope coded affinity tag. The other is SILAC, which stands for say stable isotope labeling with amino acid in cell culture. And the other is ITRAC, which is called as isobaric tag for relative and absolute quantitation. So these approaches allow for both quantitation and comparative differential proteomics. So what do I mean by quantitation? Like I said, all these techniques are used for the quantitation. So that means that although I am separating the proteins, but at the same time, from these instruments, I would like to know how much of the protein was there. I know now that this protein was there, but now I'm also interested in the quantity of the protein, because let's say I'm running two samples here. One is diseased and the other is controlled, and that is healthy. So I'll take blood sample from both of them, extract out the serum from both of them, and then I'm going to run it on this machine. So I would definitely be interested in that. Okay, yes, protein X was present in both healthy and in disease, but protein X was seen more in diseased and less in healthy, right? So these type of analysis are really useful for us in research. Otherwise, what is the point of doing all these techniques, right? So that is why I said that they are useful in both quantitation and comparative and differential proteomics. So there are also other less quantitative techniques such as MUTPIT, stands for multidimensional protein identification technology. So I'm going to do a separate video on ICAT, CELAC, iTrack, and MUTPIT. Please go check them out as well. So the SmartFit, it offers the advantages of being faster and simpler. And other gel-free chromatographic techniques are also present, for example, gas chromatography and liquid chromatography. So let's talk about mass spectrometry workflow in a very simple way. So regardless of how the protein sample is prepared, the downstream MS workflow comprises three main steps. First is the protein peptides are ionized by the ion source of the mass spectrometer, which is here. The ionizer is making the ions out of the sample you're pouring in, and then the resulting ions are separated according to their mass and charge ratio in mass analyzer, and then the ions are detected in the detector. So this is the minimum minimum instrument which go in the mass spectrometry, and you can have a little bit of changes here and there, and then different types of mass spec workflows can also be generated, which we will see in a separate video which is on mass spectrometry. So on the left hand of your screen, you can see that when we are using gel-free techniques like iTrack, CELAC, and let's say gas chromatography, liquid chromatography, you can directly enter the sample from the protein mixture into the MSMS system. But on the right hand side, you can see when you are using the gel-based techniques, first of all, you have to cut the gel and take out the protein which is embedded in the gel. Then you digest it and then you feed this sample into the mass spectrometry. So this is the difference between the gel-free technique and the gel-based techniques. So there are two main ionization sources, mainly MALDI and ESI. So MALDI stands for matrix assisted laser desorption ionization, and the other is electron spray ionization, which I have covered in my mass spectrometry video. Please check that out. So other less common sources include chemical ionization like electron impact and glow discharge ionization. So now this was all about ionization. Now let's talk about the types of analyzers. So we have like four main mass analyzer. First is time of flight, ion trap, quadrupole, Fourier transform, ion cyclotron, and electrostatic sector and magnetic sectors are the other two which are less commonly adopted mass analyzers, but they are there. So these are the four main types of mass analyzers. So now we have talked about the ionizers, and we have talked about the mass analyzers, and what is left now is the detector. So now first of all, let's talk about what is tandem MS. So like I was telling you, you can use different types of analyzers together. You can use different types of ionizers together within the MS. So when you use different types of analyzers and ionizers both at the same time during the MS protocol, it is called as tandem. So peptides can be subjected to multiple rounds of fragmentation and mass analysis, a process termed as tandem MS or MSMS. So by combining the same or different mass analyzers in tandem, such as quadrupole TOF or triple quadrupole, which is QQQMS, so the strength of the different mass analyzers, this can be leveraged to further improve the capacity of the proteome-wide analysis. So if I will apply more sensitive mass analyzers, I am going to get the definite and individual picture of the peptides which are present in my sample. So simple MS setups such as multi-top, they are used only for peptide mass measurements. Whereas if you're planning to use tandem mass spectrometers, so they are used to determine the peptide sequences as well. They are so sensitive. So let's now talk about top-down proteomics versus bottom-up proteomics. So in top-down proteomics, the proteins in a sample of interest are first separated before being individually characterized. Whereas in bottom-up proteomics, which is also called as shotgun proteomics, so all the proteins in the sample are first digested into a complex mixture of peptides. And then these peptides are then analyzed to identify which proteins were present in the sample. So, in case of top-down proteomics, first of all, the protein separation will happen with 2D dye-H or MS. And when using 2D electrophoresis technique, the proteins are first resolved on the gel and then individually digested into peptides and then analyzed by mass spectrometer. So, when the mass spectrometer directly analyzes it, the undigested sample containing the whole protein is injected into MS and the proteins are separated and individual proteins are then selected for digestion and further round of MS for the analysis of the peptide is done. So it happens again and again. But in case of shotgun proteomics, proteins are first of all digested and the digested peptide mixture is fractionated and then subjected to MS. So first of all, it will get fractionated with the help of, let's say, liquid chromatography or GC, and then this GC will be combined with MSMS. So you can either use LCMSMS or you can use GCMSMS configuration. So they'll always already be, fractionated and separated, and then this resulting peptide sequence is compared to the existing database using automated search algorithms. And then these search engine match the experimentally obtained peptide spectra from our machine to the predicted spectra of the proteins, which is produced by in silico digestion. So this is called as peptide spectrum matching. So there are several different bottom-up workflows possible, including data-dependent and data-independent methods, as well as hybrids of these. So this is the top-down and the bottom-up approach. So top-down MS is more appropriate for research on different post-translational modification and protein isoforms, wherein the shotgun technique, which is also called as bottom-up, it provides an indirect measure of the proteins which are originally present in the sample. So after this is done, we need to do the analysis of the data in proteomics as well. So proteomic studies, particularly those employing high throughput technologies, they can generate huge amount of data. And in addition to the sheer quality of the data produced by proteomic data analysis, can also be relatively complex for certain techniques such as shotgun MS. So adding to this complexity is the range of bioinformatics tools available for the proteomic analysis. So what it says is that already the data was very complex when we retrieved it from shotgun MS, and the bioinformatics tools are in turn very complex. So we need some researchers, those who are very specified and qualified in data analysis of the proteomic data. So whenever you're planning proteomic experiments, scientists they need to factor in not only the cost of the reagents and laboratory equipment, but also that of the data storage and analysis. And they have to appraise the level of the bioinformatics skills and computational resources required. So now these are the areas wherein the proteomics can find its applications: personalized medicines, biomarker discovery, drug discovery development, systems biology, agriculture, food science, paleoproteomics, which is the study of the evolution and the archaeology and the study of the ancient proteins. So that is what I was referring to, that now we have so sensitive equipments that whenever we get a sample from paleoproteomics, even if the sample is an attomolar range, we'll be able to quantify it, and astrobiology as well. So this is the future of the proteomics. So currently the proteomic workflow is relying heavily on mass spectrometry, and as powerful as this technology has proven, researchers are now looking ahead for to a future for proteomics that lies beyond the MS. So despite the attomolar sensitivity of MS detected, this implies that low concentration target molecules, example serum biomarkers, they can be detectable in a complex milieu such as human serum unless first enriched for. So Scientists are still searching for the novel high throughput proteomic techniques, which are essentially for human proteome and which can directly read entire protein sequences and identify their PTMs and does not need to draw inferences from the databases of theoretical protein matches. So that is yet to do. The science is still progressing and the research is underway. So I hope it was valuable and informative for all of you. Thanks for watching.